So early on, we have an early embryo. That's split into two, right? So maybe it was 8 cells, and then it split, so you have 4 cells, and then those cells are big enough that they can produce 2 separate embryos. Now, sometimes we have the vanishing twin. Right? So sometimes early on, they might sense that there are 2 embryos, but sometimes then later on, there is only 1. And so sometimes people actually could become, even if they are, this would be dizygotic, not identical twins. Sorry. So the vanishing twin would be like, I have a fraternal twin in utero with me, and then we have become 1. And so the vanishing twin can sometimes lead to what they call chimeras. And actually know a couple of people that are chimeras. And what a chimera means is that they have, you had 2 embryos fuse into 1. And so the extreme examples if like I had a brother in utero, and then I fused with him, then some of my cells are going to be male, and some of them are going to be female, right? And this actually can sometimes lead to quite dramatic health problems, because the problem is that your immune system might detect the one set of cells as being foreign and might actually attack it. So it could lead to some immune system disorders. And the most extreme example, or that was in the news recently, was a man who was a chimera. And the chimera part is that his twin is actually the one that contributes the sperm in his body. And so when he was DNA tested for his offspring, it was discovered that he wasn't the father, he was actually the uncle. But he didn't have a brother, right? He has no brother, right? So what that means is that his brother became part of him, and the brother is the one that contributes to the sperm. So he was really confused.
So if he reproduced, would it be incest?
No, it's not incest. It's that he was actually an uncle to his children when they tested his cells from his cheeks or blood, right? So when they went in and did that, his sperm, however, comes from his vanished twin that he incorporated into his body. So his, vanished twin was his brother, and so that was why when they looked at his offspring, it didn't pair up as being the father. Yeah. I actually know somebody also that had a vanishing twin, but they ended up as a growth on the outside of their body. So they were born with a tumor. They took the tumor off and they analyzed it, and they found that it was a twin that had just been hadn't survived.
Is that almost like a parasitic twin?
Is there such a thing in animal biology?
No, in human biology, there's a parasitic twin where the twin doesn't fully grow and the egg doesn't split and only part of their body is formed.
Oh, so is it similar to Siamese where
yeah, except they die and they don't fully form. So part of their, they could have legs hanging out their stomach.
Yes. Yeah. So that would be the parasitic. Yeah. Yeah. Okay. And obviously if they don't fully form into 2 individuals, then you could get them sharing organs, and then you get what is called Siamese twins. Okay. Okay. So we can talk about what happens when there are errors in meiosis. And particularly, some things that could happen is that the chromosomes do not separate properly. And this has a name, and it is called non-disjunction. So disjunction is separation, but the chromosomes don't separate properly, hence it's non. disjunction. The chromosomes are not separating. So they're not being pulled apart. So properly. So this can lead to what is called aneuploidy. So ploid means the number of chromosomes. U would be good number of chromosomes. And means without the correct number of chromosomes. Okay, so A means without a good number of chromosomes. So we could have trisomy. So somy means body, means chromosomes. And so this is where instead of 2 copies of a chromosome, we have 3 copies. So what is a really common, relatively common trisomy in our population? What's a common trisomy? What chromosome is commonly, where do you get an extra copy of? Down syndrome, right? So trisomy 21 is Down syndrome. Now remember that the first pair of chromosomes was really big and it had a lot of genetic material. By the time you get down to the second or the 22nd pair, they're very small, right? So there's not a lot of extra genetic material, which might explain why we can have extra first or 21st chromosome, but not an extra first. And so it's important to realize that non-disjunction could take place in your first chromosome. But if I'm a trisomy 1, I am not going to be able to survive. So I will be spontaneously aborted. If I'm trisomy 2, 3, 4, that's just too much genetic material. So it's only a few examples of trisomies that actually can survive to adulthood. Okay? You can also have monosomy. So that means that you have only 1 copy of a chromosome. But the only chromosome that you can have only 1 copy of is the sex chromosome. So this means that they have a good copy, right? This means that there's no copy. So that 0 just means that they have only 1 sex chromosome and it is an X. If you had just a Y, if you were an embryo and you had no X, you would not survive. Right? So this has a name. This would be a female, and it has a name. It's called Turner's syndrome. Okay. And so it is relatively mild aneuploid because males only have 1 copy of the X chromosome, so we really actually only need 1 copy of all the genes on the X chromosome. But it does have some effects on fertility, some effects with learning disabilities, okay, but it's not a dramatic effect. If we saw somebody with Turner syndrome, you might not even know that there was anything wrong with them. Okay, the other thing that can happen is that chromosomes can break. Or be translocated. So when you send off your genetic material to be karyotyped by a geneticist, these geneticists know exactly what they're looking for. They have a search image. They know exactly what each chromosome is supposed to look like. And if a chromosome doesn't look like that, they know almost immediately that what is wrong, right? So there is an example where you have a deletion. So this is my example. You have a deletion on chromosome number 5. So when they look at chromosome number 5, one of them is too short, and they note that. And so it would, in their karyotype analysis, it would look something like this. Okay? So a geneticist who's looking at this chromosome, for you and I, it doesn't look that much different than the normal one. but this is one is missing a piece of its arm, right? And so this is actually a disease that is not all that uncommon. It's not too common, but it's called Cri du Chat. Cri du Chat. Okay. So that actually translates in French to cry of the cat. Because these babies have abnormal vocal cords, so their vocal cords don't develop properly, and they also have characteristic facial features, and they do not develop properly, their nervous system doesn't develop properly. So this is my deleted region, okay? This is the most common syndrome caused by chromosomal deletion, so it affects between 1 and 20,000, 1 to 50,000 babies. So for some reason, the 5th chromosome is more fragile and prone to breakage. And then individuals have a characteristic phenotype, which is what they look like. So the outer expression of that deletion is that they're small at birth, they have respiratory problems, their larynx isn't developed, right? Very distinctive facial features. They have small brains, right? They have heart problems, skeletal problems, right? So that just missing that little extra bit of the 5th chromosome has dramatic effects on their phenotype, right? Okay. Okay, so today we're going to start talking about Mendelian genetics, and this is a whole different way of probably studying, because now we're going to be doing essentially math problems. And you have to practice them because it looks really easy when I am doing them up here. Some of you are going to love it because it is not straight memorization. It's more like problem solving, trying to figure something out. And some of you are probably going to hate it because it does require a little bit of practice. So when we go back to Mendelian genetics, this is in the 1850s. This is before we knew what the genetic material was. We have no idea what the DNA is. We have no idea that DNA is in chromosomes. We cannot see chromosomes. And so the first experiments were done by a person named Gregor Mendel, and he was just doing breeding experiments, putting things together, and then watching how the offspring looked and counting them. And he used pea plants because pea plants allow you to easily fertilize by taking the pollen from one plant and fertilizing it by using a paintbrush and dabbing it on the flower of another plant. And they produce a lot of offspring. The peas are actually the offspring that you can then plant, and then they'll grow into another adult, and then they'll reproduce again. So you can do many generations. Okay? So this is also prior to, we're going to talk about evolution after we get past genetics. But this is this is actually at the same time that Darwin was doing his work. But Darwin and Mendel had no communication, and they didn't know about each other at all. So what Mendel explains is how variation can be maintained from generation to generation, which is actually a really important contribution to understanding natural selection. So in pea plants, you have different traits. So for example, you could talk about the seed shape, and then you have variation in those traits. And so round or wrinkled, seed color, yellow or green, flower, purple or white. So when he was doing this, he was very smart. He decided to try to do really simple traits that have very little variation. In fact, it's discrete variation, because plants were either tall or they were dwarfed, right? They either had yellow or green pods. So there was no like in between, right? Because that would have like muddied his understanding. So it would be like if humans came in short versions and tall versions, and that you were studying height, right? Humans come in all different heights, so that's much harder to understand using simple genetics. So if we use the tall and the dwarf plants as our example, he started out with what are referred to as the parental generation. These are true breeding. And so what true breeding means is if you take their offspring from this population of plants and you plant them again, they will always have the same phenotype as their parents. They will always look the same. So sometimes this is called the P. So this is P, parental generation. So we have tall plants and we have short plants. Those are true breeding. Now, there was a theory that Mendel was trying to test, and this was the idea that you always will produce an intermediate form. So you would think that if you had tall and short, that you might get a plant that was like medium, right? So you would think that, and that was kind of the hypothesis, is that you would always get an intermediate between the two parental phenotypes, right? But when he did this, what he found is that he got tall plants. He did not get short. And so that was like, whoa, how did that happen? And then once they're tall, the idea was that then you have lost the short trait, and so it can never come back out again, right? So this is called F1, or the filial generation. Filial just means offspring. F1 generation. Right? So if we take two plants that have been produced in this way, so this means that two F1s are crossed. What do you think the F2 generation is going to look like? You might hypothesize that, well, offspring are always blended, right? So they're always a blend, so they would all be tall, right? But what he discovered is that 3/4 were tall and 1/4 was short. So somehow, even though the shortness got lost in the F1 generation, it reappears here. So what I'm talking about here with plant height, this is what we call the phenotype. So these are phenotypes. So they're the outer expression of the genes. Okay. They're the outer expression of the genes. So what we now want to talk about is, what are the genes? Now, he didn't talk to them, he didn't actually call them genes. We call them genes now. I think he called them factors or something that can be inherited from parent to offspring. So he came up with this idea that in this particular case, we have a gene. Okay, so this is a piece of DNA that codes for a protein. Now, he didn't know this, but this is what we now know. An allele is an alternate form of the gene. Right? And these alternate forms of genes, alleles, are designated by capital and lowercase letters. Okay? So let's say that we have the gene T. I'm just going to use big capital letter. This codes for height. We have alleles big T, this is the dominant allele. And this is really important. This is little t. This is the recessive allele. That's just a squiggle. So this is my recessive allele. Okay. Why didn't I put S here? Why is it lower t and not an S? Because this is alleles of gene T, right? So to put S here, that would mean that it's a separate gene. That's really important. So I put the little t here, okay? So we can talk about the genotypes, which are the combination of alleles. So you could be homozygous dominant. Homo means same, zygote. So that would be capital T, capital T. That's my genotype. Notice that I have two genes, right? These are on, we now know, chromosomes. They'd be on homologous chromosomes because homologous chromosomes have the same genes. We can have homozygous recessive. That would be little t. And then you can have the genotype heterozygous. Right? That means that you could produce gametes with either a big T or a little t. So hetero means other, homo means same. So heterozygous is always a combination of a dominant allele and a recessive allele. So if we go back up to our breeding experiment, we can now explain how you can have a tall and a short and get in the first generation all tall, but in the second generation a proportion that are short. Okay, so let me use a different color. So what do you think the genotype of this is, this one? It's true breeding. What is it? Okay, it's true breeding. Short? Homozygous recessive, little t, little t. Okay. So if we take sperm or pollen from one plant and eggs from another, what is going to be the genotype of this F1 generation? Are they going to be homozygous? Nope. They're going to be what? Heterozygous, right? Because they can only have this combination, right? We can't tell the difference between heterozygous and homozygous dominant because the dominant allele masks the expression of the recessive allele. You can't tell that it is there. So in terms of plants, those would be true breeding. What do you call that one? That is not true breeding. What's the opposite of not true breeding in plants? You see this on your seed packets. They say, this plant is, you guys know this, hybrid. Sometimes I feel like I'm speaking a foreign language. You guys are like, I have no idea what you're saying. Hybrid. So unfortunately, when I wanted to grow pea hollyhocks, right? I bought my pea hollyhocks. I didn't notice it said hybrid on them. They grew up, they flowered, they put their seeds back in the ground. The next year, I thought, oh, I don't have to plant them again because they're coming up. I'm like so excited. And then I got red and yellow hollyhocks. I did not get pink because they were hybrid. They were not true breeding, right? So purebreds versus hybrids, right? Okay, so these ones in the F generation, they could either be big T, they're tall, or big T, little t. Okay, so 3/4 of them, those we can't tell the difference by just looking at them. Okay, and these would be little t, little t. They are short. Okay. So the combination of the alleles, genotype, what they look like, their outer expression, it doesn't have to be something that you can see. It could be like your hemoglobin is your genotype. But it could, a gene that codes for defective hemoglobin, for example, the hemoglobin would be the phenotype. But that's the expression of that combination of alleles is your phenotype. Okay, so we will stop there for today. And don't forget to come to lab if you didn't come on Monday. Don't forget to do your homework on Sunday and study for your quiz Monday.
That was really bad.